Hello everyone and welcome to Space Force, where space is a driving force in ecology. Last week we discussed multi-scale habitat selection. This week we've reached our final episode, and we'll be discussing resistant surfaces and animal movement. Resistant surfaces can be defined several different ways. A resistant surface may represent the willingness, the physiological cost, reduction in survivability, or some combination of these different aspects of an animal to cross a particular environment. In other words, the resistant surface represents the cost of an animal moving across the landscape. The cost of movement can be thought of as a continuum, ranging from low resistance to high resistance. Low resistance means that there is a high movement, and high resistance means that there is low movement. Resistant surfaces are commonly used to represent connectivity in metapopulation and corridor models. In these models, isolation is described by resistance, which accounts for multiple environmental variables, instead of simply isolation by distance. There are typically 4 steps in constructing a resistant surface. The first step is to select the environmental variables that may influence the movement of the focal species. The second step is selecting the type of data, which may either be expert opinion or biological data such as detection, relocation, pathway, or genetic data. It is recommended to use empirical biological data rather than relying on expert opinion only. The third step is selecting and applying an analytical process such as a resource selection function. However, if there are other behaviors besides resource selection that drive movement, then those behaviors must be accounted for separately. Like we saw in class with our landscape resistance game, the ideal path becomes less predictable as the landscape becomes more fragmented. And finally, step 4, which is to apply resistance estimates to the selected environmental variables.
Let's illustrate this with an example. In a recent study, researchers in Southern California examined how habitat connectivity and host relatedness influence the spread of feline immunodeficiency virus in bobcats. Environmental variables included topographic roughness, vegetation density, streams, urbanization, and roads. They used empirical genetic data consisting of host phylogenetic analyses and FIV genetic sequences. Pairwise genetic distances between individual bobcats were used for resistant surface optimization for each environmental variable. Their results suggest that the spread of FIV is influenced by large-scale urban barriers to host movement. While analyses were based on resistant surface, the researchers acknowledged that a least cost analysis may have been appropriate due to the many barriers that limit bobcat movement in Southern California. While context-dependent, integrating resistant surfaces, landscape genetics, and disease ecology provided more insight into transmission dynamics within this study system.
Thanks for watching, and we hope you've enjoyed listening to Space Force.